I'm Noel Troxclair, extension entomologist based in Uvalde. I work for Texas AgriLife Extension Service. The project that Diane Rowland and I worked on, we were wanting to investigate the impact that different cropping practices and some foliar sprays might have on the physiology of plants infected with zebra chip. The bottom line of what we found is that strip tillage, the plants actually came up a couple of weeks earlier. A difference in plant growth for about a month until the later emerging caught up with them. We also looked at something called primed acclimation, and with primed acclimation, we basically challenged the plant with a deficit of water, not enough that it's really drought stressed or wilting or anything like that, but basically to stimulate the plant to upregulate certain plant metabolites, develop a more vigorous root system, and then in doing that, later the plant was better able to handle some of the stress associated with the Liberibacter infection or the zebra chip infection. The other thing that we looked at was the effect of different growth regulators. We looked at auxins, boron, and gibberellic acid. In the auxin and boron, actually, we had improved yields over the non-treated, and they seemed to handle the stress better once the plants were infected than the non-treated plants. What we found with the boron and the auxin, they also had a higher actual potato yield, and the boron treatment actually we had a more uniform potato size in addition to the increased yield, and we think that might be a management tool for getting a more uniform size from the standpoint of harvest management. One other thing, the measure that we use with PCR is called CT scores, and the plants that were infected early actually, even though they had lower CT score with more infection, they were actually better able to handle the stress early than those that were infected later.